Dear students, welcome to Chalkless class. We are delighted to have you here as we embark on a fascinating journey into one of the most fundamental and vital processes in the living world, photosynthesis. As biology students, understanding photosynthesis is absolutely essential, as it forms the very foundation of life on Earth. Every breath of oxygen you take, every morsel of food you eat, and every drop of fuel derived from organic matter can be traced back to this remarkable process occurring silently inside the green leaves around us. In today's session, we will travel through history, exploring the brilliant experiments of scientists like Priestley, Ingenhousz, von Sachs, Engelmann, and van Niel, and build a complete picture of how photosynthesis works at the cellular and molecular level. By the end of this session, you will understand not just the what, but the how and why behind this extraordinary process. So open your notebooks, stay curious, and let us begin. What is photosynthesis? Photosynthesis is defined as a physico-chemical process in which green plants use light energy to synthesize organic compounds. Two major roles are highlighted. It is the primary food source for all life on Earth. All heterotrophs ultimately depend on autotrophic plants. And it is responsible for releasing oxygen into the atmosphere, which makes aerobic life possible. Joseph Priestley, 1770. Priestley conducted experiments using a sealed bell jar. He observed that a burning candle gets extinguished and a mouse suffocates inside a sealed jar. However, when a mint plant was placed inside, the candle kept burning and the mouse stayed alive. His hypothesis was that plants restore to the air whatever breathing animals and burning candles remove. Essentially, plants recycle air. Ingenhousz, 1779. Building on Priestley's work, Ingenhousz discovered three important things. First, sunlight is essential. The plant only purified the air when placed in sunlight, not in darkness. Second, he observed small bubbles, later identified as oxygen, forming around the green parts of aquatic plants in bright sunlight, but not in the dark. Third, only the green parts of plants release oxygen. Non-green parts cannot perform this function. Two scientists made further advances. Julius von Sachs, 1854, proved that glucose, stored as starch, is produced during plant growth, showed that chlorophyll is located inside special organelles called chloroplasts, and established that green parts of plants are the site of glucose synthesis. T.W. Engelmann, 1880s, split light into spectral components using a prism, illuminated green algae in a suspension of aerobic bacteria, and found that bacteria clustered mainly in blue and red light regions, producing the first action spectrum of photosynthesis. Cornelius van Niel, 1930s. Van Niel's key insight was that photosynthesis is a light-dependent reaction where hydrogen from an oxidizable compound reduces CO2 into carbohydrates. By studying sulfur bacteria, which use H2S instead of H2O, he concluded that the oxygen released by green plants comes from water, not from CO2. The corrected overall equation he proposed is 6CO2 plus 12H2O produces C6H, 12O6 plus 6H2O, 6O2. This was later confirmed using radioisotopic techniques. Timeline of discoveries. A summary timeline of all major milestones. Priestley, 1770, showed plants restore air. Ingenhousz, 1779, found sunlight and green parts are essential for O2 release. Von Sachs, 1854, linked glucose starch production to chloroplasts. Engelmann, 1880s, determined the action spectrum. Van Niel, 1930s, proved O2 comes from water. Together, these experiments established that photosynthesis uses light, CO2, and H2O to produce carbohydrates while releasing oxygen.
For tailored topics and curated content, connect with us today. WhatsApp 8639823875.